Today we're going to continue our discussion of biochemistry, focusing on some of the major macromolecules that are so important to life. So one group of these macromolecules are the carbohydrates. So the word carbohydrate is trying to tell us something about their structure. Carbo is a reference to carbon. Hydrate means to add water. So when you look at least the simple carbohydrates, they typically have carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. So an example of that would be then, if we're looking at a carbohydrate with 6 carbons, that would translate to 12 hydrogens and 6 oxygens. Now, in terms of functions, carbohydrates have a number of important functions, but the really primary function is as a source of energy. Glucose, fructose, and starch are all examples of carbohydrates that we readily use for energy. And there are other carbohydrates that are also important for energy, but those are some that you hear a lot about. There are some carbohydrates also that are structural. For example, cellulose is a component of plant cell walls, and so again, it is part of the structure of a cell. And then also, they do play a role in immune system recognition. Our white blood cells, for example, recognizing foreign substances, sometimes look at proteins, and other times we'll look at carbohydrates. And there are some other functions for carbohydrates as well, but I think that covers the major functions. Now, when we talk about carbohydrates, we're going to follow the same formula we talked about before. There are monomer units that can be assembled to form polymers. And so our monomer units in carbohydrates are referred to as monosaccharides. And the term monosaccharide is a reference to a single sugar. That's what the word is trying to tell us. An example of this would be glucose. So glucose is an example of a carbohydrate that is also a monosaccharide. Now, when we talk about organic molecules, and I think I've mentioned this before, the structure of the molecules is really more important than the overall formula. So when we look at the formula for glucose, it's C6H12O6, following our 1 to 2 to 1 concept. And when we look at the structural formula for glucose, this is what's known as the shorthand notation. And when you look at the shorthand notation, we're supposed to be finding 6 carbons, 12 hydrogens, and 6 oxygens, but we're not seeing those. We can see 1 carbon here, we're seeing 3 hydrogens, 2 oxygens, but where are the rest of the elements? Well, in these shorthand notations, the lines are representing covalent bonds. And wherever 2 or more lines intersect, that's where a carbon should go. So if we wanted to know where the carbons go, there would be a carbon there, 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 and there. And so those are our 6 carbons. And again, this would be consistent in any kind of carbohydrate that you see drawn in the shorthand notation. Now, you'll also see some lines hanging. At the end of these hanging lines, an OH group goes there, and we call those alcohol groups in organic chemistry. So alcohol group, alcohol, alcohol, and alcohol. And then, last but not least, carbon forms 4 covalent bonds, and we can see that this carbon here has 3, for example. So extra bonds are saturated with hydrogen in organic molecules most typically, and so they normally don't even draw those bonds in. So actually there would be a hydrogen at this spot. We can see that this other carbon only had 3 bonds, so we draw in the 4th, only 3 bonds here, so let's go ahead and draw in the hydrogen. Only 3 bonds here, so draw in the hydrogen. 3 bonds here, so we need to draw in the 4th to a hydrogen. So this is the actual detailed structural formula for glucose. And now if we counted up everything, we would find our 6 carbons, we would find our 12 hydrogens, and we would find our 6 oxygens. Now let's look at another monosaccharide. Oh, and I should mention that it's helpful in figuring out these structural formulas to know the number of bonds that some of these elements form. So for example, hydrogen will form 1 covalent bond, oxygen will form 2 covalent bonds, as will sulfur because they're in the same chemical family. Nitrogen will form 3 chemical bonds. Carbon will form 4 chemical bonds. So again, that's helpful sometimes in figuring out whether you need to add a hydrogen if it hasn't been shown. Okay, another monosaccharide then is fructose. Both glucose and fructose are very common in fruits and almost in any living tissue for that matter. Now, when we look at the structure of fructose, you'll notice that the chemical formula is exactly the same, C6H12O6. So the difference in the molecules is in their structure. So actually what I would recommend is actually stopping the video and seeing if you can figure out where the carbons, the OHs, and the hydrogens would go in this particular example. But I'm going to go ahead and proceed and show you where these elements would be. So again, any place where we have 2 or more lines intersecting, that's where a carbon would go. So carbon, 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 and carbon. So that's going to give us our 6 carbons. We see the hanging lines in sugars, so we're going to put alcohol groups there. And then, again, each carbon should have 4 bonds. This one does have 4 bonds. This one does not. So we're going to have to add hydrogen, add hydrogen, add hydrogen. And now we would have our full complement of C6H12O6. Still another monosaccharide is galactose. Galactose, again, a very common sugar in nature. And you'll notice that it's very similar to the structure of glucose, except that the alcohol and hydrogen positions happen to be reversed here. So when we look at glucose, fructose, and galactose then, they all have the same chemical formula. But structurally, they're all different from one another. And so when we have organic molecules like this, and this is a pretty common situation, we refer to them as isomers. So be a little careful not to confuse the term isomer with isotopes, which we talked about earlier. But isomers then would be organic molecules with the same chemical formula, but different structural formulas. Okay, now a couple of other monosaccharides. These are structural monosaccharides. One of these is ribose and the other is deoxyribose. And you'll notice that they differ at this second carbon. Ribose has an alcohol group, whereas deoxyribose only has a hydrogen there. So deoxyribose is a deoxygenated form of ribose. And deoxyribose is an important sugar in DNA, genetic material, and ribose is an important monosaccharide in RNA, which is also a type of genetic material. Now, in addition to monosaccharides then, monosaccharides can be assembled to form disaccharides. So the prefix di means 2, saccharides mean sugar, so these would be 2 sugars bonded together. And as we're going to see in a minute, you're going to put those together through a dehydration synthesis, or that's how the body would do this. So maltose then is composed of 2 glucose molecules. Sucrose is composed of a glucose and a fructose, and lactose is composed of glucose and galactose. So let's go ahead and look at how these could be put together. So glucose and fructose, we can see OH and OH, so we want to be able to extract water from this to form a dehydration synthesis. And so those form sucrose, combination of glucose and fructose. Next, we can see 2 glucose molecules coming together, dehydration synthesis. Or I'm sorry, a glucose and a galactose forming lactose. And finally, 2 glucoses, dehydration synthesis to form maltose. So this is what our disaccharides would look like. Finally, then, we can continue to add monosaccharides to this budding chain to form polysaccharides. And we're going to be concerned with 3 polysaccharides. And I should mention that there are a lot more monosaccharides than we've discussed, a lot more disaccharides than we've discussed, and there are a lot more polysaccharides than we will discuss. However, we will look at and consider starch, glycogen, and cellulose. Now, what they have in common is that they are all 3 composed of 100% glucose, repeating units of glucose. There are a few differences, however. So starch is a way that plants store excess glucose. And so these will be in essentially straight chains or some branching chains, but not a lot of branching. So this is how plants store their excess glucose in the form of starch, 2 forms of starch. Animals will store their excess glucose in the form of glycogen. And again, it's still just long chains of glucose. It's just that there are a lot more branches to those chains, a little more complex. And again, we store those in our liver and in our muscles, and we can use the glucose for energy. Now, cellulose is a little bit different in that it's a structural carbohydrate, again, composed of 100% glucose, but they're put together in a slightly different way. And sometimes slight differences in structure translate to very different functions. So as you'll see here, in starch and glycogen, the glucoses are put together in a certain orientation. All the glucoses are in the same orientation. And in cellulose, glucose is offset. So alcohol group here and then here, so it's turned upside down every other time. And the difference is that we can digest starch. We have enzymes, molecules that will recognize and break off these starches, and we'll use those for energy. Yet our enzymes cannot recognize this configuration, and so cellulose cannot be used by animals as an energy source because we cannot recognize these individual glucoses, we can't break them off, and so we can't use them for energy. So cellulose then is just something that goes out in our fecal material. It's part of the quote-unquote roughage of our diet, and we get no energy value from cellulose. But again, it is important to plants because they use that as a cell wall. Now, proteins. Proteins are composed of amino acids. And as opposed to starch, which is a homopolymer, or glycogen or cellulose, which are all homopolymers, proteins then are heteropolymers because there are 20 different amino acids which could occupy each spot in a protein. And proteins have a number of really important functions, some of which we will get to in our next module. So one of the foundations I want to lay out initially here is that when we're talking about proteins, the amino acid sequence of the protein is crucial because that determines protein shape, and that in turn will determine protein function. So we will eventually be talking about protein function, but today we're going to focus on really protein shape. So amino acid sequence determines protein shape, determines protein function. Okay, so let's go ahead and start then with the amino acid structure. Now, all 20 amino acids have certain things in common. They will have a central carbon, and attached to that is a hydrogen. Also attached to that will be an NH2, which is an amino group, and then this COOH group, known as a carboxyl group, also known as an acid group, organic acid, because this hydrogen can dissociate and contribute to the acidity of a solution. So they're trying to tell us something about their structure in the name. Amino for amino group, acid for the carboxyl group. So again, all 20 amino acids will be exactly the same at these three positions. Where all 20 amino acids will differ is at this fourth position, where this R is. In chemistry, that's referred to as a radical group, and it just means that it can be highly variable. You can put all kinds of different things at that spot. So the 20 different amino acids will all differ at this radical position. And let's go ahead and look at some of the differences here. Now, I certainly wouldn't expect you to know these structures, but again, it's just to bring out and emphasize the kinds of things that we can see at this R position. So the simplest amino acid, glycine, just has a hydrogen there. But you can see that there can be methyl groups. There can be actually very complex, even ring structures that go into this R position. Also, I've put a green spot next to amino acids that are called essential amino acids, and I'll discuss that in a couple of minutes. So again, it's not so much the specific structures here that are important, but it's just to show you that there's a lot of variability at these R groups. And the R groups are also called side chains. Sometimes they're referred to as side chains. So again, tremendous variety within the amino acids, and there are 20 different amino acids. Now, when you put amino acids together, they again go together through a dehydration synthesis, kind of the same old thing. So again, we're looking at two amino acids that are already bound together. We have an OH and an H. They'll bond to form hydrogen, and then that will lead to the carbon bound to a nitrogen. So we refer to this link bond between amino acids as a peptide bond. The word peptide is just another term for an amino acid. So the link between the amino acids, the peptide bond, is the same. But what differs then are the side chains. And this is why the sequence of amino acids has an effect on the overall shape of the protein. So when we look at amino acids, there are 20 different amino acids. 10 of those we describe as essential amino acids. And if something is essential, of course, you have to have it. And in this case, in this chemical sense, it means that we cannot construct the essential amino acids. If we cannot construct them, that means we have to get them through our diet. So the essential amino acids must be consumed in our diet. Now, there are also 10 non-essential amino acids, which means that we can make those. So if we have the 10 essential amino acids, we can make the 10 non-essentials from the essential amino acids. So it's really not important to get all 20 amino acids in your diet. It's just important to get the 10 essential amino acids in your diet so that you can construct proteins. Now, complete proteins are those proteins that have the 10 essential amino acids. So again, the 10 essential amino acids, not all 20, just the 10 essential, because if you have the 10 essential, you can make the 10 non-essential. And all animal proteins are complete. So it doesn't matter whether you're actually eating, flesh, chicken, beef, fish, whatever, but also dairy products, milk, cheese will also have complete protein, and egg also is a complete protein. So any animal protein is a complete protein. Incomplete proteins, on the other hand, lack at least one of the 10 essential amino acids, and plant proteins, for the most part, are incomplete. So if you are a vegetarian and you want to ensure that you're getting a quote-unquote complete protein in your diet, what you need to do is combine plant proteins in a way to ingest the 10 essential amino acids. And probably the easiest combination to remember is if you combine any cereal or grain with legumes. So cereals or grains include things like wheat, corn, barley, oats, and legumes would be any member of the bean family, so beans, peas, and lentils. So that combination will give you all 10 essential amino acids. It's generally recommended that you consume those within a 24-hour period. And then you will have a satisfactory diet in regards to protein. So again, going back to this theme as it regards proteins, the amino acid sequence determines protein shape, which will determine protein function. So what are some of these protein structures then? Well, one is the primary structure of a protein. Now, ironically, the term primary structure really does not refer to a three-dimensional structure. What it's a reference to is the amino acid sequence of a protein. So if I was requesting from a researcher what is the primary structure of a protein, they wouldn't send me a three-dimensional model of the protein. What they would send me is the amino acid sequence. So perhaps it's methionine followed by leucine, followed by isoleucine, proline, alanine, glycine, and so on and so on. That's the primary structure, the amino acid sequence. It's really not showing me a three-dimensional structure. It's the sequence that is actually the structure. So that's a little hard sometimes to wrap your mind around, but that's an important idea. So primary structure is a reference to the amino acid sequence. Now, based on that amino acid sequence, some proteins will take on a secondary structure, and there are two three-dimensional structures that we refer to as secondary structures. One is an alpha helix, which is essentially a spiral, and the other is a folded composition, which we refer to as a beta pleated sheet. So if we're talking about an alpha helix, then basically you can think of a slinky. So basically a spiral-shaped protein then is an example of a secondary protein. Now, typically, the entire protein won't necessarily be spiral-shaped. Parts of it will be. And then a beta-pleated sheet, it's a reference to if you took a piece of paper and folded it up like this, it would form a beta-pleated sheet. So that's what they're trying to describe. So again, it's usually not the whole protein that is either spiral or in a pleated sheet, but portions of the protein. Now, an example of a spiral protein, however, is collagen, which connects our tissues together, bones to bones, muscles to bones, and so on. It's a connective type protein. And an example of a protein that is a beta pleated sheet would be silk proteins. So, but generally, we're talking about parts of proteins that exhibit these secondary structures. Now, tertiary structure, or a protein with a tertiary structure, is one where the secondary regions continue to fold up into a more globular type of structure. So globular proteins we refer to then as tertiary proteins or tertiary structures. Now, again, what determines or why is it that a protein will take on a secondary structure or tertiary structure? Well, it has to do with the interactions between the R groups, between the side chains. And there are a number of interactions which can occur. Some of the side chains are non-soluble, so-called hydrophobic or non-polar. So these non-polar regions then are repelled by water, forced together. Additionally, we can have hydrogen bonds, basically just attractions between slightly negative regions and slightly positive regions of our R groups or side chains. Covalent bonds can actually form between some of the side groups, R groups, particularly between sulfurs, and then also ionic bonds also can form between side chains. So these interactions then between the R groups will twist and turn and force the protein into a particular three-dimensional shape. And again, that all traces back to the initial amino acid sequence. Now, quaternary proteins are like superproteins. This is where our tertiary subunits actually come together and associate with other globular proteins or other proteins. An example of this would be hemoglobin. Hemoglobin is composed of 4 subunits, and hemoglobin is an important protein for carrying oxygen in our blood. Now, there are other factors besides primary structure that do affect protein shape. One of these would be temperature, particularly heat, and heat can cause something called denaturing. So let's consider if you cook an egg. You take a raw egg, you put it in the frying pan. The white of the egg is initially somewhat transparent. It's not perfectly clear, but it's somewhat transparent. As you add heat to that egg white, and the egg white, by the way, is virtually 100% protein, it's going to change and it's going to turn opaque and it's become a little more rubbery and so on. Now, if you take that egg off of the frying pan or out of the frying pan, or if you take it off of the stove, does the egg go back to being an uncooked egg, a raw egg? And the answer, of course, is no. So one of the things that's interesting about proteins is if you add heat, it will change the shape of the structure of the protein. But denaturing is where you permanently change the shape of that particular protein. So once you've permanently changed the shape, it can't go back. Now again, this has important implications to life. We expend a lot of energy to try to maintain a constant body temperature. And certainly if you've ever been to the doctor, if you have a fever, they normally aren't too concerned if it's a fever of maybe 100, 101, 102. But as it gets over 102 to 103, 104, there's a lot of panic which sets in. And again, the reason is that if your fever continues to spike, and this is especially true with children where fevers can change very rapidly, basically you'll cook yourself. You'll denature the proteins in your body, permanently change their shape, they won't do what they're supposed to do, and you're going to die in those situations, or potentially have severe damage, brain damage, and so on. So a little bit of temperature is not a bad thing. What we're trying to do is actually cook the organisms that are infecting us, but if we cook ourselves too much, we cook ourselves. Now, pH also affects the shape of proteins. Acidity and basicity. So again, we expend a lot of energy to maintain a constant pH in our bloodstream, and a lot of other organisms do this as well. So again, if blood gets acidic or basic, potentially that also can denature proteins. And then ultimately, what determines the amino acid sequence of a protein actually is the DNA nucleotide sequence in our genetic material. So ultimately, it's the DNA, the genetic material, that determines the sequence of amino acids. And so human DNA codes for human proteins, mouse DNA codes for mouse proteins, and so on. So I don't want to belabor the structure of chromosomes or DNA, but I do want to follow this concept through a little bit. When you look at a chromosome, the genetic component of a chromosome is DNA. And DNA is composed of repeating units known as nucleotides. So again, we're looking at this monomer-polymer concept. So again, we're going to talk more about structure of DNA later, but what I would like you to know right now is that there are nucleotides that link together to form the strands of our chromosomes, our DNA. And there are 4 different nucleotides that are used in building DNA. So DNA has a certain sequence of nucleotides that translate then to a certain sequence of amino acids. So the DNA nucleotide sequence of chromosomes determines the amino acid sequence, or the primary structure, of our proteins, which in turn determines protein shape, which in turn determines protein function. So looking at an example of this, in normal hemoglobin there's going to be a series of amino acids, a certain sequence of amino acids. Again, they'll have certain side groups, R groups, and they'll fold up into particular shapes. And in the case of hemoglobin, these beta subunits will associate with so-called alpha subunits to form a quaternary protein, which is the hemoglobin. And then they will carry oxygen in our blood. Now, a single amino acid substitution changes this beta subunit shape such that instead of being kind of a soluble molecule, the hemoglobin actually forms crystals with less surface area. And so these crystals then of hemoglobin do not efficiently carry oxygen. They also affect the shape of the red blood cell, and this is what causes sickle cell anemia. So the concept here then is that the amino acid sequence changes. That changes shape of protein, function of protein. But if we trace this back then, this change in the amino acid sequence is due to a change in the nucleotide sequence of the DNA. And so these spontaneous changes that can occur in DNA we refer to then as mutations. So at some point there was a mutation which occurred, which led to a change in the amino acid sequence and a difference in hemoglobin. So changes in the nucleotide sequence of DNA, mutations, can lead to changes in the amino acid sequence of a protein, which in turn can lead to changes in the protein shape, which in turn lead to changes in protein function. Now, the next part of that then is that nature makes a decision about these changes. The changes are potentially harmful, helpful, or neutral. And what determines whether something is harmful, helpful, or neutral essentially is if it's harmful, it makes you less able to survive and reproduce. Helpful we would define as making an organism better able to survive and reproduce. And a neutral mutation would have no effect on one's ability to survive or reproduce. So when nature is selecting for traits then, it's typically selecting for those that are either helpful or in some cases neutral. And we refer to this as natural selection. So when nature is selecting for beneficial changes in the genetic material, that's natural selection. And the way that it selects for you is that you are better able to survive and reproduce. And so now what happens is those genes build up in the population. So what happens potentially over time then is that this new genetic change, which translates to a novel protein with a beneficial outcome that builds up in the population over time, that can lead to a change in the genetic makeup in a population over time. And quite simply, that's what evolution is. So this is a concept we're obviously going to come back to, and I hope you like the way I snuck in evolution here, that when we started talking about protein shape. But again, this is a really important concept, and it's something that we will come back to several times, in that it's the genetic material that determines amino acid sequence, protein shape, and protein function. And changes in protein function then can either be harmful, helpful, or neutral. And if these help you or make you better able to survive and reproduce in a population, those genes build up in a population that changes the population genetically over time. And that's all evolution is, the change in the genetic makeup in a population over time. All right, so that does it for today, and I'll see you next time.